In the case of us, we're a bit more complex than a bacterial cell, so we have more complex cellular division. We have mitosis versus meiosis, all right? Mitosis is used very, very vaguely or very loosely in this case, all right? We refer to mitosis as being our overall cellular division, all right? But mitosis is really something more specific than that. So in this case, our mitosis cellular division is going to be a way for us to create more cells. So when your cells die, they need to create new cells. Or when you've hurt yourself and you need to heal, you create new cells to heal yourself, right? In our case, for multicellular organisms, it's going to be how we create somatic cells. Remember, somatic cells, 2N. 46 for us, all right? Somatic cells being body cells, blood cells, nerve cells, skin cells, all right? Meiosis, however, results in haploid, N, or in our case, 23, all right? This is for gametes, which are sperm and eggs, all right? Meiosis will come up again in the next unit. So, focusing on our cellular division, all right, our mitosis, our basic cellular division. We've got a first growth phase, synthesis, all right? Synthesis should be something that kind of jumps out at us, to synthesize, to make, all right? So something's going to be made during the synthesis phase. We got a second growth phase, we've got this mitosis, all right, and cytokinesis. All right, first growth phase, synthesis, and second growth phase, we can kind of lump them together into that word that I referred to before as interphase. Interphase, inter means between. Interphase is what's happening between the other stuff, basically. So, our cell cycle. All right, if you notice, G1, synthesis, G2, that's a whole lot of time before we get to the actual division. All right, the actual division chunk is that mitosis and cytokinesis. So, our interphase, our first stage of cellular division, all right, where the cell seems like it's resting. It's not really actually resting. What's happening is that in interphase, the cell is growing twice, all right? And we've got the synthesis phase. Synthesis phase is where DNA replication happens. Remember, we said synthesis is to make. What's being made is a copy of the DNA, a copy of that genetic material. So, first growth phase, cell is going to grow quickly in size. We're going to get a bigger cell. In the synthesis phase, the DNA is going to be copied, and each chromosome, all right, is going to have two chromatids attached at a centromere. And then we have our second growth phase, where our cell grows more, and the cell prepares to divide. Picture of our interphase. Then we have mitosis, PMAT, our 4 stages, which we're going to talk about in even more depth on our next benchmark. Cytokinesis, all right, is the step that follows mitosis. The step that follows mitosis is where the cytoplasm actually divides. The cell is going to pinch together, and you're going to get 2 separate cells. But it's different in plant versus animal cells. In animal cells, we have something called the cleavage furrow. Basically, the cell membrane just starts to pinch in. All right, as that pinching in takes place, so we go from one big giant cell, it starts to pinch in. All right, as that pinching in gets further and further defined, it'll eventually pinch completely into 2 cells. So what you have there, if you can see, is that we have 2 separate cells that are starting to pinch apart. Our cytokinesis in plant cells, however, doesn't have a cleavage furrow. Instead, we end up with vesicles. All right, the vesicles, remember a membrane-bound sac? Vesicles are made by the Golgi apparatus. And what they start to do is they start to line up along the midline of the cell, and eventually you're going to have a whole big line. All right, and those vesicles, those little bubbles, are actually going to come in contact with one another, and they're going to form one big giant vesicle. All right, that one big giant vesicle is then going to become a part of each separate cell. As you see here, here are those vesicles. All right, and what they form is something called a cell plate. And then that cell plate will become a boundary for a new cell. So, overall, cell division, I pet my albino tiger carefully, or I pet my angry tiger carefully. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Interphase can be broken down into 3 steps: first growth phase, synthesis, and second growth phase. Prophase, metaphase, anaphase, and telophase are also referred to as the mitosis portion. And then we have cytokinesis, where the cell divides, the cytoplasm separates. We'll continue more about this in the mitosis benchmark, so stick around.